Hello everyone, this is Kamakshi Shureka from JIS Institute of Advanced Studies and Research, Kolkata. Welcome to the webinar series 4 organized by the Center for Health Science and Technology. Thank you all for being here. I would like to take the opportunity to mention that our courses like MSc in Medical Biotechnology and Bioinformatics, Advanced Diploma in Bioinformatics and PhD programs. So admissions are going on in all those courses, and detailed information regarding all these 3 courses can be found in our website, and all contact information is given there and also visible on the screen. Email us or call us for further information. So without any further delay, I would like to move on to our talk today. And it gives me immense pleasure to welcome today's speaker, Dr. Shilpak Chatterjee. He did his master's from University of Kolkata and PhD from the Chittaranjan National Cancer Institute. For the postdoctoral study, he went to Medical University of South Carolina, Charleston, USA, after which he joined CSIR, Indian Institute of Chemical Biology, as senior scientist. He was awarded with the prestigious Wellcome Trust DBT India Alliance Intermediate Fellowship and also the American Association of Immunologists Trainee Abstract Award. Talking about the research interest, so his main research interests are immunometabolism, tumor immunology, and T-cell biology. The major objective of his lab is to understand the mechanism dampening the objective repose of the anti-tumor T cells. Today, he will be talking about cancer immunotherapy, a brilliant move to fighting the foe. I should also mention that live streaming of this talk is available on our YouTube channel, JIS IASR. And also, please write your questions to the chat section of Zoom or comment section of YouTube, which will be addressed at the end of the talk. So without any further delay, let us all welcome Dr. Shilpak Chatterjee.
Thank you for the generous introduction. So let me share my screen. Just
yes.
share with me.
We have stopped sharing ours, so hopefully you will be able. Yes, we can see it.
Is it visible now?
Yes.
Okay, so 1 second, please. Okay, so I think I'm audible. So let's start.
Yes, perfectly.
Okay, so thank you for having me today for this webinar series. And as the title suggests, so I'll be talking mainly on this cancer immunotherapy. And so first of all, what do we mean by this cancer immunotherapy? In a simple way, so the immunotherapy of cancer is just to boost up our immune system so that they can fight the cancer in a proper way. And as okay, sorry. So is it my slides are moving or no? Okay, yeah.
So yeah, now we are on the second slide.
Yeah.
Yes.
So actually, it is known that this immunotherapy of cancer, it's really has revolutionized the cancer treatment. And why it's so? So that's why I put some picture of the patient who got the immunotherapy. So if you see this patient, before the immunotherapy, he had a metastatic melanoma and the tumors are all over the body. But after this immunotherapy, almost all the tumors are gone. And similar cases for this patient also, there's a very big tumor in the patient. But after like 1 year of the immunotherapy, there is no visible tumor. So basically, all this picture, all this data suggest that this immunotherapy got the very high potential in controlling the tumor in a very good way, and so that's why it is considered as this therapy has a almost near curative potential. So it can cure the cancer most potently as compared to the other therapies. So, but how this journey started actually? So if you look back, so if you look back, so actually the journey of the immunotherapy started long back at near about 1863, when the scientist Virchow first reported that the tumor is not actually mass of malignant cells. Rather, it is composed of different other host cells, mostly the immune cells. So that time immune system was not fully discovered, and nobody knows why these host cells are doing at the tumor site. So then the crude way of immunotherapy started back in 1898 by William Coley. So he's considered as the father of immunotherapy. So what he did, he was working in a hospital in New York, Memorial Hospital in New York. So he used the Staphylococcus organism mixture, heat inactivated form, and was injecting the patient with a sarcoma. And that crude mixture was termed as the Colex toxin, and he found that the patient are responding remarkably. Okay, and he had treated like more than hundreds of patients, and obviously he didn't know why it is like in some patient why the therapy is showing some remarkable result. But he thought something is going on, something, because it was a tumor, but he was treating something with bacterial product. So he knew that something host response is going on, but he was not clear how that host response to some particular bacteria is actually driving a potent immune response or potent response against the tumor. But that time he didn't know that immune system is playing the role. But later it was discovered, yes, that toxin is actually driving the immune system. But the actual way how this immunotherapy or immune system came into the picture of this tumor biology, it started back in 1957. So when the Barnett and Thomas first proposed a mechanism or proposed a model of cancer immunosurveillance. So they showed that the immune system or immune cells actually infiltrates to the tumor site, and they have some certain roles. They are infiltrating there to stop the tumor growth, but there somehow the cancer is managing with some mechanism to evade the system and growing. But that was the piece of information first proposed by these two scientists. Yes, immune system has a major role. It is not just there, but they are playing a major role in cancer development, progression, and its outcome. Okay, and the actual immunotherapy, if you consider, and nowadays this immunotherapy is very much applicable to the patient with the bladder cancer, that is BCG. Again, BCG is not it's not a particular chemo or particular immune boosting agent for the tumor. Rather, BCG, BCG is the bacterial or the product, the vaccine. So, but that component, the BCG component, has been shown to be very useful for the bladder cancer treatment, and the role is to trigger the immune system. The BCG triggers the immune system in such a way that it is helpful for the bladder cancer patient. So after 76, like 83, 85, so these areas are considered as the like a golden era of the immune biology, because the immune system, the component of the immune system, different cell types was discovered, and people started recognizing the role of immune system in this tumor progression and tumor pathogenesis. And this way the whole story started. And most advanced immunotherapy nowadays are being used. It's actually started in between like 1985 to 2000, this area, in this time span, the modern era of immunotherapy started. So before we go to the role or how this immune system is actually working or against the tumor, so we first have to understand how our immune system works and which cell component is actually important for the anti-tumor response, because the immune system is composed of different cell types, starting from the macrophage, dendritic cells, then it comes to the adaptive immune system like T cells, B cells. But which cell type is most important to clear the tumor cells, right? So if you consider how our immune system works, so once the pathogen, or which is foreign in nature, when they enter into the body, the antigen presenting cells or the dendritic cells or macrophage, they actually engulf the pathogen and they present the antigen to their surface. And then the T cells, either it is CD8 plus T cells, we call it cytotoxic T cells, or if it is CD4 plus T cells, we call it T helper cells. So these two cell types, then they recognize the antigen when the antigen are being produced or presented by the antigen presenting cells. And once they recognize the antigen, they start proliferating or get activated. And that way they work, okay? So for tumor, it has been shown that these CD8 T cells or the cytotoxic T cells are most potent. Why? Because they have a capacity to directly recognize the tumor cells and then kill the cells. But when we talk about the immune system, or when we read our textbook, we mostly read about the antibodies. But you have to understand one thing, that in case of tumor, the antibodies are not that much efficient in killing the tumor cells directly, because antibodies are soluble in nature. They are important to neutralize the soluble pathogens in our body, but they don't have that capacity to recognize the tumor cells and directly kill them. So that's why you need T cells, mostly the cytotoxic T cells or CD8 T cells, to control the tumor. So as you see here, that the activation process of CD8 T cells require an antigen, right? So the question is, do the tumors express the antigen? The answer is yes, they do. There are two important types of antigen. One is called the tumor neoantigen, and this antigen actually they don't present in the normal cells, but they start expressing on the tumor cells when they become transformed. So that's why these neoantigens are very much potent in inducing the immune response. And there is another set of antigen, we call it altered self antigen. So what is altered self antigen? So these antigens are present in the normal cell types, but their expression level is very low. And when the got transformed and when they become the cancerous, those protein got overexpressed or get overexpressed. So that's why they can be used as a antigen. So basically, that means that the tumor, they have the antigen that can be recognized by our immune system, and our immune system can be activated by seeing this antigen. So there is two antigen, one is neoantigen and another one is altered self antigen. So if you see that how this immune recognition happens in our body, so it's called the cancer immunity cycle. So what happens? So suppose there is a tumor growth, right? And if you see here, if there is a tumor growth, the tumor growth actually attract the component of the innate immune system to the site of tumor. So what are those components? One is the major component is the dendritic cells. Another major component is the macrophage. So what they do at the tumor site, they actually carry the antigen. They actually engulf the tumor cells. They actually carry the antigen, and then they go to the nearby lymph node where the lymphocytes are residing. Okay, so lymphocytes means T lymphocytes and B lymphocytes. So once these dendritic cells or antigen presenting cells carrying the antigen and then go to this draining lymph node, they actually present this antigen to the residing T cells. And this T cell, after seeing the antigen, they become activated. They then recruit to the site of tumor and they start killing the tumor. So the point is that, so if this cancer immunity cycle is active in our body and it is active in every individual, and ideally in any individual who has a active immune system, they should not develop any tumor. But we do. Why it is so? Because the tumor somehow finds some mechanism to evade the killing by these anti-tumor T cells. So this was actually shown or proposed by a very eminent scientist. So what they have shown, that there is a 3-step by which the tumor cells modulate the immune system in a way so that our immune system don't react to the tumor cell. So what is the way? What is the 3-step? So they call it 3 E's of cancer. 3 E's. What is the first E? First E is elimination. So what is elimination? So when there is a tumor cell, when there is a transformation happens to the normal tissue, and these cells become transformed or become cancerous. As soon as they become cancerous, our immune system can recognize that the immune system infiltrate to the site of inflammation or the site of cancer, and they start killing the cancer. Okay, if the immune system is potent enough in killing all the transformed cells, then tumor will not develop. But what happened after this killing and elimination process, there are some tumor cells which actually escape the killing by the immune system. Okay, and they enter into the stage of equilibrium. So in the equilibrium stage, there is a continuous killing by the immune system to the tumor cells. But there are certain type of cancer cells, they actually escape this killing and they slowly start growing at the site of cancer or at that site. And then the most important step happens, which is the escape state. What is the escape? So by slowly growing, the tumor cells find some mechanism to block or to hinder the killing process of the immune cells. Okay, and they start recruiting other immune cells which are actually suppressive in nature. Suppressive in nature means this Treg and MDSC, they are also T cells and the component of the immune cells, but their job is not to kill the tumor. Their job is to suppress which are actually killing the tumor. Their job is to suppress these CD8 T cells or NK cells which are actually acting against the tumor cells. So once this stage is attained, then tumor start grows. Okay, and we see the visible tumor or the clinical tumor, and the patient go to the clinic. So once the patient reach to the clinic, you have to understand that the tumor has attained this step or this stage, which is called the escape stage, where they actually recruited some suppressive cells like Treg and MDSC, which is suppressing the anti-tumor T cells or anti-tumor NK cells. So what is the mechanism? How this Treg and MDSC suppress our immune system? So if you search in the PubMed, you will probably find, so I searched in PubMed, I found there are 5,000, more than 5,000 paper which actually described how this Treg suppress the anti-tumor T cells. So the Treg is nothing but a particular type of CD4 T cells, and they express a particular or unique transcription factor called FOXP3. So Treg can be recognized by their expression of CD4 and FOXP3, and it's a T cells. Okay, and the role of this T cell is to find a myriad of mechanism to suppress the anti-tumor T cells. And same as for the MDSC. So MDSC is a myeloid cell. It's called the myeloid derived suppressor cells. So these are myeloid in origin, and they also use the sort of similar mechanism to suppress the effector T cells. So as a result, when these two cell types like Treg and MDSC is present, effector T cells cannot perform their job. So scientists thought, okay, let's deplete this Treg and MDSC. So there is a depletion antibody. So if you deplete specifically Treg and MDSC from the site of tumor, then probably you can boost up your immune system or boost up your anti-tumor T cells, right? So they did that, but there is a problem. So what is the problem? The problem is that this same Treg is actually important for maintaining the immune homeostasis. So what is immune homeostasis? So actually in every individual, we have some autoreactive T cells. So autoreactive T cells means those T cells are against our self antigen. So during our normal thymic development, those T cells are produced and they are in the periphery. But our system anticipated that thing, and that's why they made Treg and MDSC. And the role of the Treg and MDSC in healthy individual is to keep those autoreactive T cells in check so that they can avoid the collateral damage, right? Because if you deplete Treg or MDSC, you can unleash those autoreactive T cells, and those autoreactive T cells will damage all the organs, and it's a very fatal disease. So they tried depleting the Treg and MDSC, but found that depletion of Treg and MDSC is not a very practical way to boost up the anti-tumor response. You can boost up, but at the same time, you can or you will induce autoimmune disease in the patient when you deplete Treg and MDSC. Okay. So the question is that then why or how can be this anti-tumor effector response can be achieved? So they found one thing when they were doing this kind of experiment, depleting Treg and blocking their suppressive mechanism. They found one thing, that if you remove this kind of suppressive mechanism from the tumor site, you can actually boost up the T cell response, but not to the extent of resolving the tumor. Okay, so then they realized, yes, immune suppression is there in the tumor microenvironment, but maybe there are some mechanism by which cancer is also making the T cell dysfunctional. So it is not just the suppression by the Treg or MDSC, rather there is some mechanism by which the T cells are becoming dysfunctional when they are going or when they are reaching to the tumor site. So to understand that dysfunction, so we have to first understand what is the functional state, how the T cells become functional. So initially it was thought, or initially it was discovered that to activate the T cells, you need one thing. What is that? You need a particular antigen which can be recognized by this TCR or the T cell receptor. So the T cell receptor is very unique. It is unique for particular antigen, and once the antigen is there, the T cell can recognize that antigen and the T cells can activate it. But there is a catch. What is that? For the T cells, you need the antigen to be presented by a particular MHC. MHC means the major histocompatibility complex. So if the antigen is not presented by the MHC, then the T cells cannot recognize the antigen. So that means the antigen has to be presented by the MHC. So there are two types of MHC, MHC class 1 and MHC class 2. MHC class 1 present the antigen to the CD8 T cells, and MHC class 2 present the antigen to the CD4 T cells, right? So once this recognition happens, once the TCR recognize the antigen presented by the MHC, they start proliferating. So T cells become clonally proliferate and make lots of T cells. And these dots, what I have shown, trying to show here, these green dots and red dots, these are basically the cytokines or effector molecules secreted by the activated T cells, and these molecules are the important factor which actually cause cell lysis or tumor cell lysis and anything. But then scientists have realized that only this interaction is not sufficient. So they soon realize that, yes, you need TCR recognition of the antigen presented by the MHC, but along with this signal, you need another signal, which is called the signal 2. So this is the model called the signal 1, signal 2 model of T cell activation. So what is the signal 2? Signal 2 is that there are certain receptor present on the activated T cells. We call it co-receptors, okay, or the co-stimulatory receptor. So this like here I'm showing one co-stimulatory receptor like CD28. So the role of CD28 is to recognize the another ligand present on the antigen presenting cells. So once this ligand receptor interaction happens along with the recognition of TCR of this antigen, then the optimum activation and proliferation of the T cell happens. Okay. So after realizing this thing, scientists started working on this field. So what are the different co-stimulatory receptor present on the T cell? Is it the only CD28 or there are certain others? So it turned out that there are certain quite handful of molecules are present on the T cells which can be used as a co-stimulatory receptors, right? One is CD28, ICOS, OX14, and all these things. But the point is that the major discovery in the field of T cell biology towards the development of modern immunotherapy started with these two gentlemen. And these two gentlemen, James Allison and Tasuku Honjo, they got the Nobel Prize in 2018 for their discovery of very important feature of T cell biology. What is that? They identified that, yes, you need these co-stimulatory molecules for the T cell activation. But what they identified the breaks on the T cells. So they identified that there are certain molecules also present on the activated T cells. They are not co-stimulatory, rather they are co-inhibitory. And the important molecules they discovered is CTLA4 and PD1. So PD1 was discovered by this Honjo and CTLA4 by James Allison. So what is the importance of this molecule? Why this is so important? Why this discovery moved this field so much forward towards the development of immunotherapy? Because of this picture. So now you can understand a car. Somebody is driving a car without a brake, and you can anticipate the consequences, right? But the car with a brake, you can stop the car at any point of time, and you can accelerate the car at any point of time if you have a control over the car. But the tumor cells actually, so what they found, the tumor cell is so clever that they use this braking technology to hinder the immune response. Okay, so what the tumor cells they do, so what the tumor cells, so actually before I go to that part, so basically these two molecules, one is CTLA4 and another one is PD1. So they actually act as a brake for the T cells, but they work in a two different way. Their mechanism of action is different. So in case of CTLA4, how it shut down the function of T cells. So suppose this is a normal T cells, they are seeing the antigen through the TCR, they are co-stimulatory ligand or receptor is bound to the ligand, and the T cells are in the activated condition. But as soon as the cells start expressing the CTLA4, the CTLA4 has a major or has higher affinity towards this ligand which actually binds to the CD28. That's the same ligand which binds with the CD28 binds with the CTLA4, but the binding affinity of this ligand towards CTLA4 is higher, and as a result it actually transduce a negative signal to the T cells, and T cell shut off their effector function. But in case of PD1, it actually doesn't hamper the binding of CD28 with its ligand. Rather, PD1 has its separate ligand. It's called PDL1 or PDL2. And once this interaction happens, this actually shut off the effector function of the T cells. So these are two different mechanisms how this PD1 and CTLA4 work. So tumor, as I was telling, the tumor is so clever. They use this technology, or they use this strategy to shut off the anti-tumor T cell response. So what they do, so suppose it's a normal T cells, which is effector in nature. They are seeing that antigen, tumor antigen, their CD28 is engaged with the ligand called B7, and these cells are secreting all these effector molecules like perforin, granzyme B, interferon gamma, and these are actually lysing the tumor cells. So you will have a proper tumor clearance. But in the tumor microenvironment, when we call it T cell dysfunctional, what do we mean? These T cells start expressing these negative regulatory molecules like PD1 and CTLA4. And once this receptor binds with the ligand like PDL1 or the same ligand which is binds with the CD28, they actually shut off this effector function. You can compare between these two pictures. The repeat perforin, granzyme B, and the interferon gamma level is drastically reduced, and they fail to control the tumor. As a result, tumor growth. So what is the major discovery of these two guys, this James Allison and Honjo? Obviously, they discovered this braking system on the T cells. And they also suggested that if you can interfere the binding of PDL1 with the PD1, or interfere the binding of the CTLA4 with this ligand, you can probably rejuvenate these dysfunctional T cells and can achieve the tumor clearance. So what they did, they actually use the antibody which can block or which can bind with this PD1 and block the interaction with the PDL1. At the similar way, you can use the anti-CTLA4 antibody which can block the CTLA4 interaction with this B7 molecule. As a result, CD28 can get its own ligand, TCR can see its antigen, and this cell starts secreting the site lighting molecules, and the tumor can be properly cured, or tumor lysis can happen, right? So this is the strategy. It's called the immune checkpoint blockade therapy. This is the most successful therapy so far. Lots of people are getting this kind of therapy, and the success rate is remarkable. Okay. So once this strategy was being developed, there was another strategy which is called the adaptive T cell therapy. So this strategy is equally important. This strategy is equally useful in treating the patient, and in fact, a lot of patients are getting this adaptive T cell therapy treatment. So what is the adaptive T cell therapy? So they thought, okay, suppose in the tumor site you have 1,000 cells, and not necessarily all the 1,000 cells are defective or dysfunctional. You will always find some cells which still can retain the effector function. So the scientists thought, okay, say there is 1,000 cells, if we can make it million, if you somehow can make the cells number from 1,000 to million, there will be a substantial number of T cells present in the tumor site which are not dysfunctional, but they can carry the effector function and can kill the tumor cells, like the tumor cell. So what they did, they actually take out the T cells from the cancer bearing patient. They modified the T cells outside in the in vitro system, and they engineered the cells so that these T cells can recognize the tumor. They put a particular receptor, call it TCR receptor or CAR T cell receptor, and expand the cells in a huge number and put the cells back to the same patient. The idea was that the cells number is very high, and then they can kill the tumor. And this is the actual strategy how the protocol is designed. So this is the antigen reactive T cells. They actually clone the TCR region from these reactive T cells. They put it into the viral vector. They transduce the normal T cells with this viral vector, so that the normal T cells start expressing the anti-tumor, the TCR, which can recognize the tumor antigen. They expand the T cells and can put back the T cells to the same patient. So this is the strategy for the TCR engineered T cells, and there is another brilliant strategy called the CAR T-cell therapy. What is CAR? CAR is chimeric antigen receptor. So one drawback for the TCR T-cell is that, so as I discussed earlier, that T-cells always require MHC to recognize the antigen. So without MHC, T-cell cannot recognize the antigen. But in case of antibody, they don't require any MHC, say though they don't have any MHC restriction. So what they thought, if we can identify the antibody which is against the tumor antigen, then we can put the antibody to the T cell surface, and then the T cells will not require any MHC restriction to recognize the tumor cells, because in the tumor cells you will always find there is a down regulation of the MHC, and if there is a down regulation, then the T cells cannot do. So what they did, they took the antibody fragment, this fragment called the FAC fragment. So they took the FAC fragment, which is actually important for recognize the antigen, and then they fused this FAC fragment with the CD3 domain, which is actually the downstream signaling domain of the TCL receptor or TCR receptor, and in that way you can make the T cells expressing antibody FAC fragment, and so that the T cells don't have any MHC restriction, they can go and directly recognize the tumor. Okay, and there is a different version of the CAR called the first generation CAR, second generation CAR, and third generation CAR. So they just modified the CAR. In the second generation CAR, they put some co-stimulatory molecules so that when the T cells is recognizing the antigen, they are transducing the signal. So the same signal can activate the CD28 or 41BB. And the third generation, they put two co-stimulatory molecules together so that the activation signal, and it's very robust, and the T cell can fight back very easily to the tumor. But despite all this story, all these things, still the responding rate to the immunotherapy is still to 30%, and majority of the patient don't respond to the immunotherapy. Okay, so here I can I put some references, like this is the high responder group. Here the objective response rate is 87 to 70%, but you can see there is an intermediate response group or the low response group where the response rate is 25% to 20 to 25%, even more less than 25%. So the point is that immunotherapy is very potent, very active, but still there are some issue to be resolved so that we can make the immunotherapy better. Okay, so what is that? What is those part? What is that thing which is important for like making the immunotherapy less applicable to 70% of the patient? So it has been shown that dysfunctionality, it's not a stable state. Okay, so there is a dynamics in the dysfunctionality of the T cells. So when the dysfunctionality started, the cells remains in the TX means T exhausted. Okay, so the T exhausted progenitor 1 state, then they go from progenitor 1 to progenitor 2, then to the intermediate state, and then they go to this terminally exhausted T cells. Okay, so it has been shown that these three state of the T cells, they actually can respond to the PD-1 and anti-CTLA4 therapy. So if the T cells are in this state of dysfunctionality or exhaustion, you can still revert back those T cells to the functional stage. But if this stage attained, that is the terminally exhausted state, then they stop responding to the immunotherapy. And it has been shown that it is nicely correlated with the metabolic state of the T cells. And metabolism plays a very important role in regulating the effector function of the T cells. So as I was mentioning earlier, these T cells are secreting these interferon gamma, granzyme B, perforin. These all the effector molecule secretion depends on the metabolic fitness of the T cells. If the T cells are not metabolically fit, this T cell cannot respond or cannot stay longer at the tumor site. And it has been shown that when the cells are in the responding state, their metabolism is high, they are metabolically active. But as long as they enter to the terminally exhausted state, they are becoming metabolically unfit, so that they cannot respond, or they don't respond to this therapy. And why it is so? Because the tumor microenvironment is very much deprived of the essential nutrient, because the nutrient that is required for the T cell development or T cell effector function, like glucose, glutamine, tryptophan, all these nutrient is actually taken up by the neighboring cancer cells, because the cancer cell number is very high. They're taken up all the nutrient, so there is very less nutrient available for the T cells to maintain their effector function. So what happened when the T cells don't see the anti or don't see the metabolic component? So if there is a metabolic stress in the tumor microenvironment, they actually alter the signaling state, and that alteration alter the metabolic or cause the metabolic perturbation in the T cells, which ultimately lead to the epigenetic modification and altered gene expression. As a result, the T cells lose their effector function, but start expressing these negative regulatory molecules in a highest concentration. So our lab is majorly focusing on this part. We are trying to understand how this metabolic perturbation, or how to make the T cell fit, metabolically fit at the tumor site. So major question in our lab is to understand how to improve the metabolic fitness of the anti-tumor T cells. So we actually we are doing this kind of study. When we started working on this field, these two paper actually caught our eyes. So we have shown that this IL-12, which is an important cytokine present or important cytokine for T cell activation, if the T cells are primed with IL-12, they actually do better at the tumor site. So, but the mechanism was not known, and we wanted to understand how IL-12 is causing T cell functionality and make the T cells potent in terms of anti-tumor response. Okay, so we developed a in vitro system. What we do, we actually take the splenic T cells from the normal mice. Okay, and we culture the T cells, we activate the T cells. There is a certain protocol for activation of the T cells, and and during the activation, we put either IL-2 or IL-12. IL-2 is important for T cell proliferation, and our hypothesis or experimental strategy, we used IL-12, okay, instead of IL-2. So IL-2 is usually used for T cell proliferation. So when I was discussing about the CAR T cell expansion or the TCR bearing T cell expansion, they usually expand the T cell in presence of IL-2. But we thought what IL-12 is doing, let's see. And after this activation of the T cells, we put these T cells to a condition where the metabolism, or where the metabolic component like glucose, glutamines are severely depleted, and we call it TCM, tumor conditioned media. And after the exposure to this media, we actually analyze their function. Okay, so we thought what we found that when IL-2 cells were exposed to this tumor conditioned media, they completely lose their effector function. Here you can see this interferon gamma production by these IL-2 cells. Here it is 60% when the cells were not treated with this 80% tumor conditioned media. But as soon as they enter into the tumor conditioned media, the 60% interferon gamma becomes 20%. But in case of IL-12, there is no such decrease, right? Here you can see the statistics. And we used the antigen specific T cells. Here we saw the same result, that IL-2 cannot maintain the effector cytokine production in the tumor conditioned media, but IL-12 does. Okay, and we also used the human T cells. These human T cells were exposed to the ascitic fluid which was obtained from the ovarian tumor bearing patient, and IL-2 are not able to maintain their cytokine production. 21% becomes 11%, but IL-2 does. Okay, and not only the interferon gamma production, but if you see the cytolytic assay, which is important for the cytolytic, means the direct cell killing. So we activated the T cells, we then co-cultured the T cells with the tumor cells, and in case of IL-2, there is a severe decrease of the tumor killing activity when the cells were in the 80% tumor condition media, but that was not evident in case of IL-12. Okay. So to understand what is the mechanism, we did a RNA-seq analysis using with the help of Dr. Shondeep Pal, who is in the JIS University, and he helped us to understand or analyze the data. So what we, I'll just go through this data. What we found that there is a certain difference between the IL-2 cells and the IL-12 cells in terms of their metabolic activity. Okay, so our transcriptomic study led us to understand that these IL-12 cells had, if you look at these genes, so these IL-12 cells, they are more glycolytic as compared to the IL-2 cells. And also these IL-12 cells, they actually use their fatty acid metabolism when they are entering to the site or when they are seeing this nutrient deprivation like 80% TCM. But same upregulation of fatty acid metabolism was not evident in case of IL-2 cells. So we got two important lead. One is the IL-12 are more glycolytic, and IL-12 cells also can induce their fatty acid metabolism when they are in the nutrient deprived state. Okay, so then we found what IL-12 or what glycolysis is doing. So we first checked the glycolytic potential. So there is a certain way to check the glycolytic activity of the cells. We measured the glucose uptake. We found glucose uptake is more in the IL-12 cells. We used the seahorse analysis, and we found that yes, seahorse, it's a real-time flux analysis of the glycolysis, and we found that yes, glycolysis is higher in the IL-12 cells as compared to the IL-2 cells. And also the gene expression of all the glycolytic genes are more in the IL-12 cells as compared to IL-2 cells. So then we blocked all the pathways of the glycolysis. We blocked hexokinase, we blocked GAPDH, we blocked pyruvate kinase, and in all the cases we found that if you block these enzymes in the IL-12 cells, you lose the cytokine production. So this is without blockage, this is with the blocker. Okay, so here the cells are producing 51%. If you block this pathway, it becomes 17. Here 56 to 10%. So in all the cases, if you block these enzymes in the IL-12 cells, they lose their ability to control the tumor or control or produce the cytokine. Then we found that whether this transport of pyruvate, so the ultimate product of the glycolysis is pyruvate, right? So pyruvate has two fate, it can make lactate or it can enter into the acetyl TCA cycle. So what we thought which pathway is important. So we blocked pyruvate transport to the mitochondria using a specific blocker, and at the same time we block the gluconeogenesis. And when we block this pathway, again we found that if we block pyruvate entry into the mitochondria, you lose the cytokine production by these IL-12 cells. So this is without, this is with blocker, it becomes 12% of the 56%. So this data suggests that yes, the glycolysis, starting from glycolysis to the pyruvate or acetyl or pyruvate and enter into the TCA cycle is important for IL-12 cells to maintain their cytokine. Okay, and then we did the metabolomics analysis to identify which metabolite is actually doing all these things. And I'm not going through all the details, but I just wanted to point out that what we found that this acetyl-CoA level is very much high in the IL-12 cells as compared to the IL-2 cells, and which is also measured by the biochemical way. Yes, acetyl-CoA is higher in the IL-12 cells. So the acetyl-CoA is a very important metabolite because it can, it is a rate limiting metabolite to start the fatty acid metabolism, and it also can act as a major substrate for this epigenetic modification. Okay, so we thought whether this acetyl-CoA which are getting produced in the mitochondria, that is important, or the acetyl-CoA which is produced in the cytosol, that is important. So acetyl-CoA which is produced in the mitochondria, that can be converted to the citrate, and citrate can come out to the cytosol and become acetyl-CoA again by the use of this enzyme called ACLY. So we block this ACLY enzyme, and we found that yes, blocking ACLY actually, so this is the ACLY. So we first measured the ACLY gene expression. We found ACLY gene expression is higher in the IL-12 cells as compared to IL-2. And when we block the ACLY expression, we found that these cells lose their ability to secrete the interferon gamma, these IL-12 cells. So see, this is 71% cells are producing, but as soon as you block ACLY using the inhibitor, you'll lose the cytokine production by these IL-12 cells. And not only the cytokine production, but at the same time, these cells lose their viability. So this is the live dead staining. So without the inhibitor, the cells were 72% viable cells, but with the inhibitor, the cells become 27%. But this is not the case in case of IL-2. So it is not like that the inhibitor is as such toxic, but it is toxic for the IL-12 cells when they are in the tumor condition media. If you block this pathway, then it is becoming toxic for the cells. So then we found out the mechanism that it is actually important for the fatty acid metabolism. So we found that yes, this fatty acid metabolism is higher in the IL-12 cells because of the acetyl-CoA level. And if you block this fatty acid breakdown using the orlistat, so the cells become less viable and their ATP production is less. So the overall, so then another piece of data we have. So overall, what we thought that okay, IL-12 is very much costly. Okay, we cannot use in normal settings. So can we use some other strategy to replace the IL-12? So as I said, the IL-12 is mainly driving the TCA cycle citrate production, and that citrate is coming out to the cytosol and becoming acetyl-CoA, and that acetyl-CoA is doing everything. So what we thought, instead of complete media, can we feed the normal T cells with the 5 millimolar pyruvate, so that pyruvate can directly go into the TCA cycle, convert it to the citrate, that citrate can come out to the cytosol and becoming acetyl-CoA and do these things. So to prove this, we measured the acetyl-CoA level after feeding the cells with the high amount of pyruvate, and we found that yes, acetyl-CoA level is very high after feeding the cells with the this is IL-2 cells with the pyruvate. So this was not IL-12 culture, rather it was IL-2 culture, but the cells were fed with the high amount of pyruvate. And then we found that if you fed the cells with high amount of pyruvate, they are more resistant to lose their interferon gamma production. So this is the normal IL-2 cells. As I showed earlier, the normal IL-2 cells cannot retain their interferon gamma production. The 30% become 8%. But the same kind of cells, when they are cultured with high amount of pyruvate, they can retain their interferon gamma production, right? And they also, it is also dependent on their cytosolic acetyl-CoA, because when we block with the BMS or the ACLY inhibitor, we lose their functionality. And also this ATP production is important, or this fatty acid oxidation is important to maintain their viability. So to further check whether these cells, what the cells we are making, whether they are really potent in controlling the tumor, we did an anti-tumor experiment. So what we did, we used the anti-tumor T cells, which we call OT1 T cells. So these T cells carrying the TCR, which can specifically recognize the OVA antigen, which is overexpressed on the EL4 tumor. And then we injected the T cells into the EL4 OVA-bearing mice. And you can see this is a control. These mice didn't get any kind of T cells. The tumor grew in these mice. And these T cells, these mice only got the normal IL-2 culture T cells. They were controlling the tumor, but after some point, the tumor started growing into those mice. But these IL-12 cells and IL-2 cells fed with high concentration of pyruvate, they are very much potent in controlling the tumor. And we also found that these cells are actually there in the tumor-bearing host. So overall, what we have found, that glucose is important for the IL-12 cells. So what they do with the glucose, they actually convert the glucose to the pyruvate. Pyruvate enter to the mitochondria, and then they become a citrate, and that citrate comes out of the cytosol, and using ACLY, it becomes acetyl-CoA. And that acetyl-CoA maintain the epigenetic modification of the system, and also it is important for maintaining the cytokine production. And what is the role of this fatty acid oxidation that was mentioning? This fatty acid oxidation is important because when these cells are in the nutrient deprived condition, like tumor microenvironment, when the glucose is very limiting, they use their stored fatty acid and break down that fatty acid using the lipolysis pathway, and use that fatty acid derived energy to sustain in the tumor microenvironment, which is otherwise metabolically very stressed, and all the nutrients are very much depleted. So for this kind of cells, they don't require outside nutrient in the tumor microenvironment. Rather, they can use their own stored lipid and use this and break down this lipid and make the energy for the survival. So the end, what I want to say that when we are kid, or when we were studying the immunology and cancer immunology, so there is a term that cancer has no answer, but with this advantage of the T cells, probably we can remove the no from there, and certainly we will have some answer to the cancer. So with that, I would like to acknowledge my team. I have a small team, and these fellows are working very hard to make everything possible. And we are currently supported by CSIR Fund and Allcome Trust DBT Alliance Fund, and also through ACRB. And I would like to take the name of Dr. Sandeep Paul, who helped us a lot in analyzing the data of RNA-seq, the data I showed from our lab, and also Dr. Shomen Bosak and my postdoc mentor, Dr. Shikhar Marota, who is also helping us in sending some reagent and all these things. So with that, I would like to thank everyone, and thank you. So I'll be happy if you have any question. Thank you.
Thank you, Dr. Chatterjee, for such an illuminating talk. Yeah, I guess the students would be specifically benefited by this explanation and this groundbreaking research that you are doing in your lab. So, so right now,
so that's why I thought that there will be more students. So I thought keep this presentation in that form, yes, so that they can understand. But I put some data from our lab so that they can understand what this actual theory or actual story is going. So anyway,
right.
Question I'll be happy to answer that.
Yeah, so that is super exciting. So before going to the questions, so I have few questions. Can I ask now?
Sure.
Yeah. So the overactivation of the glycolytic pathway. So do you think, is there any relation with the oxygen availability in the tumor microenvironment with that?
Yeah, actually, so the strategy that we used, so we know that the tumor microenvironment, when in the tumor microenvironment, the T cells will not get enough nutrient for that effector function. So we actually use the strategy of adaptive T cell therapy. So in the adaptive T cell therapy, what they do, they actually extend the tumor reactive T cells outside the host in the tissue culture facility, and then put those T cells back. So during that expansion, we can modulate their metabolism, we can modulate their other effector function, and make the cell in such a way so that they can sustain longer in the tumor microenvironment. So that is our strategy, how to modulate their the metabolic function, so that when we put the cells back to the tumor bearing host, they can sustain there. So that's why we use the IL-12 technology or IL-12 strategy, and we found that if you put the IL-12 during the T cell activation, they actually boost up their glycolytic activity, and that glycolytic activity is important for making lipid, and that lipid gets stored in the cells. Now, when you are taking the cells from the culture and putting back to the host bearing the tumor, then the stored lipid in the cells can be utilized by the lipolysis pathway and can give the energy for their sustenance and the effector function.
Right. Thank you. So I can see questions are coming up, so let's move on to that. So the first question is from Nirabhu Chakraborty. Sir, is it possible to tabulate all the autoimmune T cells of an individual using genetic analysis? such that Tregs can be freely depleted? And hypothetically, using such a method in combination with traditional methods, is it possible to obtain a higher response?
So depleting Treg is almost impossible. So actually, you cannot map back, or you cannot map all the autoreactive T cells. So suppose you map all the autoreactive T cells, right? But so to keep those autoreactive T cells in check, you need Treg always. There is a certain disease called scurvy, and the disease is a mutation in the FOXP3 gene. Okay, so the patient with such disease, they don't survive much longer. Their survival is for few months after the birth, because of the severe autoimmunity. And because of this scurvy disease, the FOXP3 is inactive. That means Treg is gone. You will not find any Treg in those individuals. And because there is no Treg in those individuals, you cannot control those autoreactive T cells. So even if you map the autoreactive T cells by the CRISPR technology or other technology, what are the autoreactive T cells present in the body, but still you need Treg always to keep those T cells in check. But because as soon as you deplete Treg, they will be activated and they will start killing the host. But what the scientists, or even in our lab, what we are doing, we are trying to find out what is the difference between the normal Treg, which is present in the normal individual, versus the Treg which is present at the tumor site. So there is a difference. There are certain phenotypic difference, functional difference between the normal Treg and the tumor induced Treg. And now if you can target those molecules which is only present on the tumor induced Treg, probably you can deplete only those Treg which are present in the tumor microenvironment, and in that way you can boost up the immune system. So that is the strategy, the potent strategy. Also we are trying in our lab, and also it is being studied across the world, different labs across the world.
All right, so next question is from Shujan Chaudhary. Thanks for your lucid and nice presentation. It would be nice if you can put some light on how your findings will help the drug developer to improve the immunotherapy strategy. Thanks.
Yeah, so actually we are currently what we are working at, we are working the second. So I discussed two strategy. One is the immune checkpoint blockade therapy, and another one is the adaptive T cell therapy. So we are currently working on the adaptive T cell therapy. And what we have found, because CAR T cell therapy and adaptive T cell therapy, for both this therapy, you need to expand the T cells before you put back those T cells into the patient. And that expansion state is very much crucial. If you don't expand in a proper way, those T cells will not sustain longer in the patient. So people are actually trying to develop different strategy for this expansion. So what we found that IL-12 could be a potent strategy for expansion of these CAR T cells or the TCR T cells before they put back into the patient. But the another important point that I'd like to mention here is that we found that since IL-12 is very much costly, and then you need all this GMP certification and all these things before you use the IL-12 for clinical use. So that's why we found out that normal pyruvate, which is a component of the normal media, normal RPMI media that you use for the tissue culture. So that pyruvate, if you use at higher concentration, that pyruvate itself can act like IL-12 culture T cells. So in that way, you can reduce the cost in a drastically, because IL-2 is very much costly. So if you just add higher concentration of pyruvate in the culture, it will act as act exactly as IL-12 that we have found in our system, and that's why we did the tumor experiment on all other studies. So that way we are thinking we can add some value to the existing knowledge and towards betterment of the immunotherapy in a cheaper way.
So many people are thanking you for such an Wonderful presentation. Thank you, sir. Thank you, sir. I have so the chat box is actually flooded with thank you messages. So looking for questions. There's one. Is it really a great, sir, is it possible to deploy a cocktail of IL-2 and IL-12 together? By Ribu Devnath.
Yeah, actually, so people have tried injecting IL-12 in the patient. So there's a 2-3 clinical trials have been made, have been performed across the world. So IL-12 as such is very much toxic. So you cannot inject IL-12 as such to the patient bearing tumor. It is not feasible. It is not a good way because it's toxic. So what people are doing, and there are 2 trials, there is very successful trial. What they have done, they actually use some delivery system or delivery approaches to specifically deliver the IL-12 to the tumor site. Okay, and that tumor residing macrophages and dendritic cells can engulf those delivery that vesicle, and then they can start expressing the IL-12, start producing IL-12, and they can help the nearby T cells. So that is one approach. And another approach, they use I think they use this overexpression system, but overexpression system is not it is a costly affair for treating the patient. So in that way, injecting cocktail or those kind of thing will not be a viable method, a viable strategy to move forward. But obviously, this thing can be used during the expansion strategy of the CAR T cells, or as discussing earlier, expansion of the CAR T cells or the adaptive TCR T cells before it can be transferred to the patient. So, but yes, cocktail, injecting cocktail directly to the patient is not a feasible idea because it is a toxic.
Okay, so next question from Vidhi Agarwal. My question is, as you mentioned, the T cells are adapted in a way that they can undergo lipolysis pathway to enhance their metabolic activity in tumor environment. So does that imply that this therapy could potentially not be implemented for brain tumor, as we know that the brain metabolism does not favor burning fatty acids to provide energy?
Yeah, that is a very good question. That is a very good point, because for adaptive T cell therapy, you have to cross this blood-brain barrier, and I think there are one CAR T cell not trial, but two papers came out where they use these CAR T cells for certain glioblastoma, which is close to this blood-brain barrier. But again, your point is well taken, because obviously in the brain, this strategy might not work, but hopefully it can work in other tumor settings like melanoma or the other form of tumor like glioblastoma, the bladder cancer and other cancer, ovarian cancer. So we actually are working more on this ovarian cancer because of the 2 things. One is ovarian cancer are poor responder to this immune checkpoint blockade therapy. Truly or honestly, we are not working on glioblastoma, and we are right. Maybe this kind of approach where these T cells are using their lipolysis to sustain or to maintain their energy metabolism will not be feasible in case of glioblastoma. But we are mostly working on the ovarian cancer and melanoma because they are still not very good responder of the immunotherapy, and at least where immunotherapy is being tried, and they are kind of trying to improve the response rate. So currently we are not working on that field, but rather in these 2 tumors. But yes, you're right. It could be difficult. It could be difficult in treating the glioblastoma or other kind of brain tumor.
So, I don't see any more questions coming, then we might meet. So
Okay. Yeah.
if there are any more questions, so I want to thank you again on the behalf of the entire JISR family for presenting this exciting talk. And thank you so much. Yeah, the thank you messages are still coming for you. Everybody is thanking you.
Okay. Thank you.
Okay.
Yeah.
Thank you. Yeah. So with this note, I'm ending this meeting.